In this video, we will be discussing about carbohydrates. So one thing to note before we begin is that the complete notes of the carbohydrate have been given in the description box for your reference. So in this video, we will be covering the basics of the carbohydrate such as monosaccharides, oligosaccharides, polysaccharides such as starch, cellulose, etc. And the remaining portion of the carbohydrate such as retrogradation of starch, gelatinization, etc. will be covered in the second video. So let us begin by understanding what actually carbohydrates are. So carbohydrates contain carbon, hydrogen and oxygen. So if we look at an example given over here, which is of glucose, we see that the structure contains carbon, hydrogen and oxygen. Now carbohydrates are also called as saccharides, which means sugar or hydrates of carbon. As we can see from the general formula for carbohydrates, CmH2n H2O whole n, that they are basically the hydrates of carbon. And they are also the main source of energy for the body. So basically 1 gram of carbohydrates provide us with 4 kilocalories of energy, which is the second highest after fat. 1 gram of fat provides 9 kilocalories of energy. So now if you look at the definition of carbohydrates, it says carbohydrates are polyhydroxyaldehyde or ketones and their derivatives. Now if you look at these terms, it can seem very difficult, but if you try to break and understand its meaning, then it might become easier for us. So poly actually means many, and hydroxy here refers to the presence of hydroxyl group along with aldehyde or a ketone group. So let us look at what these actually are. So just for a quick revision, hydroxyl group is OH group and carbonyl group is a C double bond O. A ketone group is when a carbonyl group has 2 R attached to it, and aldehyde is when a carbonyl group has 1 R and 1 H attached to it. So if you look at, for an example, if you look at glucose, which we have just seen as an example, we see that there are many OH group attached to it. So this makes it polyhydroxy, and this glucose is an example of a sugar containing an aldehyde group. So we see that there is an aldehyde group present over here where we have C double bond O carbonyl group, and with the carbonyl we have H on one side and the R group on other side. So there is a presence of aldehyde sugar. So this makes the glucose as an aldose, and if there is a presence of ketone group, we can call it as ketose. So now if you look at the classification of carbohydrates, carbohydrates can be divided based on the number of the sugar or the saccharide units as simple or complex. So in the simple, we have monosaccharides and disaccharides, and as the name suggests, mono means one and saccharide meaning sugar. Similarly, di meaning two and saccharide again meaning sugar. Poly means many and saccharide again means sugar units. So basically monosaccharides will be containing only one sugar unit, whereas a disaccharide will be containing two sugar units joined together by glycosidic bond, and polysaccharide will be containing many sugar units joined together. So basically many monosaccharides will join together to form polysaccharides. So this becomes polysaccharide. So similarly, monosaccharide will be containing only one sugar unit and disaccharide will be containing two sugar units. So now if you look at the example of monosaccharides, we have glucose, fructose and galactose, and disaccharides we have maltose, sucrose, lactose. Polysaccharides we have starch, fibers and glycogen. So first, monosaccharides. So monosaccharides are the simplest group of carbohydrates since they are containing only one sugar unit and they cannot be further hydrolyzed. Definitely, since they are only one single sugar unit, they cannot be further hydrolyzed, and the number of the carbon atom can range from 3 to 7. So 3 will be the least number of carbons the monosaccharides can contain, and they will be known as triose. For all other names, you can refer the notes. So common monosaccharides are glucose, fructose, galactose, and based on the functional group which is attached to monosaccharide, they can be called as ketoses or aldoses, as we have discussed that they can contain an aldehyde group, as an example over here where we have a carbonyl group, we have H, and on the other side we have a R group. So they can be containing either an aldehyde group or a ketose group. For example, this one where this carbonyl group has one R group attached over here and one R group attached over here. So basically this becomes a ketose, right? So it can either be having an aldose or it can be having a ketose. So now we talk about glyceraldehyde. As we have just discussed that among monosaccharides, they can have from 3 to 7 carbons, with 3 being the least and called as triose. So glyceraldehyde is an example of triose which contains only 3 carbons. So if we look at the structure of glyceraldehyde, so it is the simplest aldehyde sugar, and if we look at the structure, we see that the second carbon over here is an asymmetric carbon. So what we mean by asymmetric carbon is that when all the 4 groups which are attached to the carbon are different, then we call it as asymmetric carbon. For example, the second carbon of the glyceraldehyde. So one side we have an OH group, on the other side we have a H group, on one side we have a aldehyde group, and on the other side we have a R group. So all the 4 groups attached to this particular carbon over here are different. This makes this carbon as a asymmetric carbon. Now in the monosaccharides, we can have pentoses or hexoses, that is basically the monosaccharide can exist in both chain as well as the ring forms. So what we see over here is an example of the chain form, and what we see is an example of the ring form over here. So glucose can exist as both in chain form as well as the ring form. Now in the ring forms, they can further be of 2 types, pyranose or furanose. Now pyranose is a 5 carbon and 1 oxygen structure which is hexagon in shape. Similarly, furanose has 4 carbon and 1 oxygen and is pentagon in shape. For example, if you look at this example, it becomes easier to understand. Here we have alpha D glucopyranose and alpha D fructofuranose. So this gluco is coming from glucose and pyranose is representing the hexagon shape of the ring. As we can see over here, it contains 5 carbons and 1 oxygen, right? Just like we discussed that pyranose will be containing 5 carbon and 1 oxygen and a hexagon shape. So in case of glucopyranose, we can see 5 carbons and 1 oxygen and a hexagon shape. Now we know where this name glucopyranose is coming from. Gluco is coming from glucose and pyranose from the shape of the ring. Similarly, we have fructofuranose, where fructo represents fructose and furanose represents the shape of the ring. It contains, similarly, it will contain 4 carbons and it has 1 oxygen. So D-glucose can be represented as D-glucopyranose and fructose can be represented as D-fructofuranose. So we will look at the meaning of this alpha and D, what they mean and what they represent in the coming slides. So now looking at what anomers are. So anomers differ in their configuration, particularly at the acetyl or the hemiacetyl carbon. Now what we mean by hemiacetyl or acetyl carbon is, hemiacetyl carbon is the carbon to which 1 OR and 1 OH group are attached to the same carbon. Similarly, acetyl is when to the same carbon 2 OR group are attached. We call it as acetyl group. So when we talk about anomers, we particularly look at the carbon which is either a hemiacetyl or acetyl. For example, this alpha and beta glucose, we are looking at these carbons which are hemiacetals because one side they contain an OH group and one side they contain an OR group. Now, alpha forms are the one where OH of C1 and CH2OH of C5 are on the opposite sides. So if you look over here, C1, here we have the OH group which is pointing downwards, and on this C5 of the CH2OH, the OH group is present on the other side. So the OH groups over here are on the opposite side. Such a form is called as alpha glucose. Similarly, if they are on the same side, we call it as beta glucose, like here. The carbon one over here, which is a hemiacetal, has a OH group, and this OH group is present they are present on the same side. Therefore, this is called as beta glucose. Now we look at what isomers are. So isomers are compound having the same molecular formula but different spatial arrangements. For example, if you look at the example of glucose, mannose and galactose over here, all 3 of them have the same molecular formula, which is C6H12O6. However, if I look at the spatial arrangement, they differ from one another. For example, if I compare mannose to glucose, we see that this carbon number 2 over here, they differ at the C2 configuration. the OH position is different and so is the H. So their spatial arrangement of the OH and H is different at the C2 or carbon 2. Similarly, if we compare galactose to glucose, we see that there is a difference at carbon 4 over here. So there is difference in the spatial arrangement of the OH and the H group over here. However, all the three have the same molecular formula but differ from one another in terms of the spatial arrangement. Such kind of compounds are known as isomers. Now we go to the next one, which is enantiomers, or we can also call it as DLN isomers. So DLN isomers are actually mirror images of each other. So when the hydroxyl group of an asymmetric carbon farthest from the carbonyl group is on the left side, it is of the L series, while if it is on the right side, it is of the D series. So let us understand this by an example so that it becomes easier. So what the definition says is that when the hydroxyl group of the asymmetric carbon, so we have to look for the asymmetric carbon which is farthest from the carbonyl group. So the carbonyl group is present over here, C double bond O, and if we look at the carbon which is farthest from this carbonyl group, it is this carbon. However, this carbon is not an asymmetric carbon since there are two H attached to it. Just for your quick revision, we have just seen asymmetric carbon is carbon to which all the four group attached are different. And here there are two H attached. So this makes this carbon which is, it is not asymmetric. So we look at this carbon. This carbon is farthest from the carbonyl group and is also asymmetric. It has H on one side, CH2OH on the other side, OH on other side, and on the fourth side there is a different R group attached. So if you look at the OH position of this carbon, which is asymmetric and also farthest from the carbonyl group, it is on the left side. Such configuration is known as L-glucose. Similarly, again, if we compare this D-glucose, if you look at the carbonyl group, again the asymmetric carbon which is farthest from the carbonyl group is this one. So the OH position is on the right. Such a configuration is known as D-glucose. So they are enantiomers, or basically they are mirror images of one another. Now let us understand what reducing sugars are. So any sugar which contains a hemiacetal group is a reducing sugar. So we have already seen before, but just for a quick revision, hemiacetal group is when to the same carbon we have one OR and one OH group attached, whereas the acetyl group is when to a same carbon we have two OR group attached. So any sugar which has an acetyl group is non-reducing, and any sugar which has a hemiacetal group is a reducing sugar. So for the reducing agents, the common tests which are used are Benedict's reagent or Felling's reagent, and the monosaccharides are classified as reducing sugar because they reduce Cu2 plus to Cu plus, which forms a red precipitate. Now, if we talk about the reducing sugar examples, it will definitely include the monosaccharides such as glucose because they have a hemiacetal group. It will also include disaccharides such as lactose and maltose because they also have a hemiacetal group. However, sucrose, even though it is a disaccharide, it is a non-reducing sugar because it contains an acetyl group. So let us look at the structures to make it even more clear. If you look at the example of glucose over here, we have a hemiacetal group over here, where we have on this carbon, we have one OH group attached and one O, and we can consider this as R. So there is one OH group attached and one OR group attached, which makes it a hemiacetal. As a result, it is a reducing sugar. Similarly, we look at the example of fructose. To this hemiacetal carbon over here, we have one side OH and one side OR group attached. However, if we look at the example of sucrose over here, we have this carbon over here is an acetyl carbon because there is O, and we can consider this as R. Similarly, O, and this can be considered as R. So basically this carbon has two side OR group attached to it, which makes it a acetyl carbon. As a result, sucrose is a non-reducing sugar. Now going on to oligosaccharides. So oligosaccharides are formed by polymerization of N molecules of monosaccharides. They contain 2 to 10 sugar units. So basically around 2 to 10 monosaccharides will join total 10 monosaccharides can join to form oligosaccharides. So commonly found oligosaccharides include sugar, maltose, lactose, raffinose and stachyose, etc. So basically disaccharides are formed or polysaccharides etc. are formed by the glycosidic linkages. Now what these glycosidic linkages are, they are basically those in which the carbon of one monosaccharide molecule in a disaccharide is bonded to other carbon atom through a O bond. As we can see over here, these two monosaccharide units are joined together by an O atom, oxygen atom, to form one disaccharide. Now, this happens by elimination of water in the process. So let us look at this glucose and glucose. So here one glucose is joining another saccharide of glucose to form maltose. Now if we look at the formation over here, these two OH groups will eliminate water. So as a result, one OH and one more H will be removed, leaving us with one oxygen atom, which will form this bond, which is known as glycosidic linkage or glycosidic bond. So now we know that once two saccharides combine, there is elimination of water to form this glycosidic linkage which connects two saccharides or two sugar units. Now sucrose is similarly formed by a glycosidic bond between glucose and fructose, or we can also call them as glucopyranose and fructofuranose depending upon the ring shape. So sucrose is composed of glucopyranosyl and beta-D-fructofuranose unit linked head to head as we have seen over here, and it is a non-reducing sugar. We have already seen that for the reason that this carbon over here is an acetyl carbon, since there is a OR and again OR group attached to it, and it is commonly called as table sugar. The second disaccharide we're looking at is maltose. So maltose is a reducing sugar commonly known as malt sugar, and it is produced by the hydrolysis of starch using enzyme beta amylase. Now one important thing to note over here is that in GATE 2015 there was a question which asked which is the enzyme which hydrolyzes starch to maltose, for which the correct answer was beta amylase. So this becomes very important. So two molecules of glucose are joined together by alpha 1,4 glycosidic bond. As we can see over here, there is again a glycosidic linkage where one unit of glucose, as we have just seen in the example of the glycosidic bond formation, that two glucose units join together and they form maltose. So the last disaccharide we are looking at is lactose. Now lactose is again a reducing sugar, and it is commonly known as milk sugar for the obvious reason that lactose is present in very high quantity in the milk, and it consists of a beta galactose with a beta 1,4 glycosidic linkage to a beta glucose. So basically one galactose and one glucose unit join together in order to form lactose. Now in the GATE 2017, there was a question to identify the correct pair of mono, di and trisaccharides. You can find the list of all the major ones in the notes. Now going on to the polysaccharides. So most of the carbohydrates in nature occur as polysaccharides, and it is a polymer of monosaccharide units joined together by glycosidic bonds, as we have just seen that they are formed by glycosidic bonds. So they are also called glycans, and they can further be of two types: homopolysaccharide or heteropolysaccharide. Now as the name suggests, homo meaning same and hetero meaning different. So once the same saccharide units join together, they are known as homopolysaccharide. For example, starch or cellulose, where only one kind of saccharide, which is glucose, joined together to form starch or cellulose. Heteropolysaccharide, where many different kinds of saccharides join together to form them, for example, pectin or gums. Now looking at the first example of the polysaccharide, we have starch. So starch is the principal storage polysaccharide of plants, and it is a polysaccharide of glucose, as I have just told that the glucose units join together to form starch. Now they consist of amylose and amylopectin. Amylose is the linear structure which consists of 1,4 glycosidic linkages, alpha 1,4 glycosidic linkages, and amylopectin is the branch structure which consists of alpha 1,6 linkages as well and has a bush-like structure. So basically Amylose is the linear component of the starch and amylopectin is the branched component of the starch. This linearity is due to the presence of alpha 1,4 linkage and this branching is present due to alpha 1,6 linkage. So if we look at the structure over here, it becomes even more clear. What we see over here is an example of amylose where we see glucose units are joined together by glycosidic linkages and they can be up to 300 to 600 units, and over here we have alpha 1,4 glycosidic bonds. And over here we have alpha 1,4 glycosidic bonds. However, if you look at the structure of amylopectin, so we have the linear component which is forming the alpha 1,4 linkages. Apart from that, there is also branching which is occurring due to alpha 1,6 linkage. As we see, carbon 6 over here is joining to carbon 1 of this saccharide, which is causing this alpha 1,6 linkage leading to branching. So amylopectin is the branched component and this branching is occurring due to alpha 1,6 glycosidic bond, and this linear part is like amylose. Similarly, 1,4 linkages. Now coming on to cellulose, which is a second example of the polysaccharide that we are discussing. Cellulose is the most abundant carbohydrate in nature and it is a fibrous polysaccharide and forms cell wall in plants. It is an unbranched structure and contains linear chains of beta glucose units which are joined together by glycosidic 1,4 linkages. Now you will be thinking that even the linkages in case of the starch was 1,4 and similarly in cellulose 1,4. Now cellulose is also formed by glucose and starch is also formed from the glucose. So what is the difference? So there is difference in this bonding. In case of cellulose, if you look over the structure over here, we have beta 1,4 linkages, whereas in case of amylose, we have alpha 1,4 linkages. Now that is the reason they differ in the structure, and this makes cellulose not soluble in water and not digested by humans. Human digestive juices lack the enzyme called cellulase, which hydrolyzes beta 1,4 glycosidic linkage. As we discussed that these bonds are the beta 1,4 glycosidic linkage and not the alpha 1,4 glycosidic linkage. So as a result, we need a special enzyme called as cellulase to digest the cellulose, which is absent in humans. As a result, human beings cannot digest cellulose. However, herbivorous animals such as cow, goat or termites, they have this enzyme cellulase within their digestive tracts, and as a result, they have the ability to digest cellulose. So cellulose differs from starch in this bonding, whereas we have beta 1,4 over here and we have alpha 1,4 over here. So with this, we end the topic of carbohydrates. The remaining portion of the carbohydrates will be covered in the next video.